So my name is Kendall Villalobos, and I am an undergraduate at the College of New Jersey. This summer, I worked in the Lee Lab, and I'll be presenting on my poster investigating possible sex determination genes in HD-ZIP and TCP1 in hornworts. So you may be wondering, what are hornworts, right? Most people do. Hornworts are actually a small group of non-vascular land plants that comprise around 220 species. Yet their unique biology offers important insights into plant evolution. Approximately 40% of all hornwort species are dioicous, meaning that they have separate male and female individuals. Dioicy in bryophytes, aka non-vascular plants, is expressed by sex chromosomes, which have sex-determining regions that carry sex-determining factors, including those genes essential for sex determination. One cool thing about these hornworts are that they have 4 autosomes and 1 sex chromosome, V designating male, U designating female. 2 transcription factors, HD-ZIP and TCP1, are found in both male and female sex chromosomes, and they each form a reciprocal monophyletic group. Adding Anthraceros angustus, a species that is yet to be sampled, helps us determine questions like the evolution of sex chromosomes, UV sex chromosomes, and the factors by which sex is determined. We did this by collecting male and female Anthraceros angustus samples for DNA and RNA extraction. These samples were then compared to concentrations of greater than 10 nanograms per microliter and 20 nanograms per microliter for Illumina sequencing. Transcriptomes were assembled using RNA spades. BUSCO and BLAST were used to assess protein set completeness and to determine TCP and HD-ZIP genes. These genes were aligned using MAFT, and then the phylogeny was reconstructed using IQ-tree and 300 bootstraps. This leads us to our gene tree reconstruction, and both trees, TCP1 and HD-ZIP, are RNA-based, and they tell us a similar relationship, right? Highlighted are the colors to see that these are the same species within the different clades, the male-associated clade and the female-associated clade, and they have their respective chromosome to denote as well. The different thing about this is that the added Anthraceros angustus transcripts that we added, both male and female samples group within the female clade, which is different from the other samples that we've seen. Some of these things have us hypothesize a few different things. Some of them that the presence of particular genes such as HD-ZIP and TCP1 do not necessarily determine femaleness or maleness. These genes may not even be sex-determining factors, or they may act in conjunction with other genes. So both of them, in addition to maybe an unknown gene, can determine sex. TCP1 and HD-ZIP may function as sex-determining factors in other hornwort species, maybe not Anthraceros angustus. The other ones group quite well. Anthraceros angustus, as you can see, does not group as well. As far as future directions, when we obtain DNA sequencing data, we can assemble genomes and we can make phylogenetic trees using that data. And we can also identify additional homologs from different sampling points and compare their phylogenetic relationships. And then lastly, as well, this is all genomic data. And sometimes we want to see what it actually looks like in real life, what it looks like under a microscope. So we can clone, transform, and overexpress Anthraceros angustus genes within the model hornwort Anthraceros angustus to observe the phenotypic differences. Are they all male? Are they all female if you overexpress them?
Super interesting. So can you tell me a little bit more about that last point that you were talking about, what they look like? Do you have any hypotheses of what that might look like under a microscope?
Yes. So actually we were doing this in lab. However, there were a few complications, but one of our hypotheses for these is if we overexpress TCP1 or HD-ZIP, what we'll see as phenotypic differences is on the plate that they're all on. When we look at them in a microscope, if you overexpress them, they will either be all female or all male, right? Because they are sex determining. So if you overexpress them, they may lead to one of them over the other.
Super, super interesting. Yeah, thank you so much, Kendall, for sharing with us. This is really great. Thank you.